We're going to go through a lot of these proteins and give new names to them. But first, we can categorize them as integral and peripheral. So integral, like integrate within, actually extend all the way through the membrane. If you think peripheral, like your periphery on the edges, peripheral proteins are just on the outside. And so depending on whether or not you are an integral or a peripheral protein, you do different functions, obviously. That's the major theme of biology, is different structure, different function. And so we have this protein extends all the way through in this picture. We refer to this as an integral protein. Here's those cholesterol bits given its shape and structure. Here's our phospholipids. Here's our extracellular matrix, intracellular matrix, inside and outside the cell. Integral and peripheral. Not so rough, right? Well, now we're going to go through different types. Channel proteins are probably the easiest to remember. They create a channel. It's selective, not just everything can flow free. Because think back to what we talked about in chapter 1 and 2 a little bit. This phospholipid in chapter 3, really, this phospholipid barrier allowed us to compartmentalize and allowed us to have organization and allowed us to separate what was outside the cell and inside the cell. That's extremely important. You can have control over what comes in and what goes out. Some things diffuse readily. Oxygen, carbon dioxide, water can diffuse right through this phospholipid barrier, but bigger molecules cannot. And you want to have control of that. You don't want all of your inner contents flooding out or things just bursting into the cell. But sometimes you need to bulk transport. You need things to move into the cell that won't fit through that phospholipid barrier. And so you create a channel, like a bridge over a stream or something. It's a channel. There's no energy involved here. Things move across what we call a concentration gradient, but certain ions and molecules can flow over that channel. Usually the channels will be named based on what happens: calcium channels, potassium channels. And so they will have a function or a name based on their function. Carrier proteins are a little different. They receive a particular molecule and then change shape to allow that molecule to come in or out. They're not just freely open all the time. And so one of the properties of life is maintaining balance, right? What was the biology term for maintaining inner balance? Homeostasis. An extremely important aspect of homeostasis is ion concentration, salt concentration inside of our cells. There's a certain concentration we have to keep of various things to allow our muscles to work, to allow cell signaling, to allow our brain to communicate to various things. And so we release these ions and then have to replenish, or vice versa. One of the extremely important ion complexes here is the sodium-potassium pump. It's how your nerves talk to each other and talk to the rest of your body, by releasing one and building up the other. And so they're interactive. One goes out, one goes in. Your cells are always leaky to potassium. And so the way to memorize the ion that your cell should have inside of it, one of the older terms, was to think of the Circle K ranch. You think Circle K, potassium is K. You have to keep potassium inside the cell. You have to. Potassium is always leaking out, constantly. It's important. It keeps the difference in membrane potential, difference in electrocharge. But potassium is always leaking out, so we got to pump it back in all the time. Sodium is constantly being forced into our cells, so we have to pump it out all the time. And so we do so in this various mechanism that we'll get into more detail later, through the same carrier protein. At one point, we're pumping potassium back in. We have enough, it changes shape, we pump sodium out. Have enough of that, changes shape, pump potassium in. It's this back and forth pump. Here is our sugar fingerprint, I call it. Cell recognition proteins. Cell recognized proteins. Cell recognition. It's how your body recognizes your cell are you. And other things aren't you, like pathogens, like a bacteria or something. Because it recognizes your ID code on the outside of your cells. It knows, your body knows your MHC complex, your glycoproteins. It's just special sugars attached to proteins out there, and it's kind of like your little ID calling card. Sometimes your body forgets, and that's perhaps how you could develop an autoimmune disorder, like lupus or something, that your body could just forget that you are you, and it begins to attack you. Or you have a situation where your body is just overactive and attacking everything, and part of that is your glycoproteins. Or you could have an invader, like a bacteria, and they have a different MHC complex than you do. And so your body knows right away, we got to get that out, that's not us. So they send out their white blood cells. But cell recognition proteins are how your cells know your cells are you. They're different for each person. I think we began to talk a little bit about that in one of the chapters. It's why we have such difficulty with organ transplant. Maybe we can match blood type as best as we can, match genetics as best as we can. But your MHC complex, your cell recognition proteins, aren't going to perfectly match somebody else. Sometimes not even identical twins. And so even though you get close, sometimes the person that has an organ, a new organ from somebody else, or even if one is grown or replaced, have to take immunosuppressants because their body constantly sees that as, hey, that's not them. And that's just your body's natural way of trying to fight off bacteria, but in the process, it tries to fight off these organs. We're getting much better about it. I imagine in the next couple of decades, we'll find a way to just know your MHC complex, put that on those organs, and away you go. I imagine in the future, we'll, instead of finding organs from other people, we'll just be able to grow your own organs from your cells. We're very, very close to that type of technology, so it'll be kind of exciting. Because that has major implications for anything, heart attack, stroke, you could just replace the organ pretty much. So anyhow, with these proteins, we also have receptor proteins. So they receive something. They're usually shaped for a very specific molecule. So we need to ask ourselves why. Because remember, sometimes it's easier to recall things if you can think about the why behind it. Why would we need a special shape? Let's think of an example. Right now, I'm already hungry. I don't know why, but I already am. And so my body is probably dropping in blood sugar amounts. If I went and ate something really fast, had a snack, that's a lot of blood sugar that I would increase. If I had too much, my body would release insulin and said, hey, we need to bring some of that sugar inside of the cells. Well, the insulin is released in the pancreas. The cells are pretty far away from that. And so you kind of just release that hormone and hope it makes it to the right cells, right? Well, if you release a special hormone like that in your pancreas or kidney, wherever we're talking about, brain, release a hormone and it's trying to get somewhere, you don't want all these cells along the way that don't need that information absorbing that hormone. And so the cells that need these hormones have a special shape, a special protein, like a lock and a key that will fit these molecules. And once they hit it, then they can bring it inside the cell. And so we call them receptor proteins. It's a major way that organs depend on signaling. It's why you can have organs so far apart in the body and they still can send a signal to the other part and things do what they're supposed to do. But they're called receptor proteins. They can be turned on and off, and crazily, you're constantly removing them and making new ones. And so you could have perfect hormone receptors one year, and then 5 years later have issues with these receptors. Because our bodies are crazy things that can do what they want sometimes. And so there are all kinds of issues that can happen with these receptor proteins, with being turned on, turned off, making more, making less. Because if you think of your body, it tries to be energy efficient. And so you don't want enzymes turned on all the time, or you don't want receptor proteins just active all the time if you don't need them for a moment. And so if we're not using something, we tend to turn it off or get rid of it and make more when we do need it. Which leads me to my next statement, enzymes. Enzymes just speed up reactions, and most enzymes are proteins. So we have enzymatic proteins as well that catalyze reactions. Now, when we think reaction and metabolism, remember we talked about catabolism and anabolism, creating something or degrading something, synthesizing or degrading, bringing things together, breaking things apart. Somebody's got to be doing that. And a major component of all these reactions are enzymes. A lot of what occurs inside of our cells would take far longer than could keep us alive if we didn't have enzymes to do it. We talked about how lactase degrades lactose. So usually an enzyme ends in ase, and these days we try to name it after what it does, so it's not as confusing to you. We kind of talked a little bit about cellulose, how we can't really break down cellulose without our bacteria. We don't have cellulase. Interestingly enough, we don't. Other primates do, chimpanzees do, but we don't because our major diet isn't all food or plant-based stuff. We have an omnivorous diet. We can eat both. We break down some, but not all. Ever eaten corn or spinach or a bunch of plant material and then you see it later? We can't break it down. We don't have the cellulase. That's why we get what we can, but some's left over. Look at this big word, adenylate cyclase. We won't use that word very much, but adenylate cyclase is important for ATP metabolism. Junction proteins form a junction. That's as simple as it is. They form a junction in between cells. Tight junctions adhere cells. Well, why would you want a tight junction adhering cells? Well, we're talking about how, well, one, let's think about our skin, right? It's our body's largest organ. All these different cell types working together to act like a giant organ. And it's kind of a barrier. It helps us maintain homeostasis, helps us maintain our temperature, helps us maintain our fluid regulation, helps us maintain all kinds of stuff. And it's protective, right? We don't just absorb everything we touch. And so we want to make sure that these cells are adhered close together. You don't want materials getting around and behind your cells. You don't want to get inside your organs. You know, you want it to be a tight junction. And so it's like a glue cementing these two cells together. Some junctions actually connect the plasma membrane to the cytoskeleton. And look at this word, you've seen it before. Peptidoglycan is the junction between plant cell walls. Where else do we see peptidoglycan? Who has a cell wall made up of peptidoglycan? Fungus are made up of chitin. Plants are made up of cellulose. Who else has a cell wall? Do we have a cell wall? No, we do not. We have plasma membrane. Bacteria, their cell wall is made up of peptidoglycan.